the longer term organization. Thank you, Walter. I'll start again so it can be recorded. Welcome all to Stability Hub office hours. We're a branch of the Pharmaceutical Stability Discussion Group, which has been around for about 35 years or more, as kind of bringing people together to talk about stability. And in this particular format, we're online and we're got a distinguished panel to answer questions and talk a little bit about their fields of expertise. But most of all, we want to entertain your questions, and we'll prioritize those. And then if those run dry for any periods of time, we'll be injecting some other topics for you to listen to. So you're welcome to spend the entire time with us. We hope you will. But if you need to get off or even get off and get back on again, that's okay. A couple of the ground rules. We need our participants to be identified in some way, shape, or form that we could get back to you if necessary. I know some of you have some rather cryptic sign-ons there. So if you can change to a recognizable first name or something along those lines, that'd be great. And also, if you can activate your cameras, it's much nicer to look into your eyes rather than just your posting there. We're well aware of people in their pajamas and cats walking through and dogs barking and little kids getting lunch. We all survived the pandemic. Half of us were in our pajamas for those years, so that's totally fine. Not a big problem for us at all. We'd like to ask if you have a question, you would try submitting it in the chat. We'll keep an eye on that to see what you have. If it's more complicated, you're welcome to raise your hand and unmute and explain your question a little bit further. Or if you have something you'd like to say about a particular topic that's under discussion, you can raise your hand and then we'll recognize you and unmute as well. We'd ask that for any session, whether you're a panelist or a person presenting a question, that we try to go in about 5-minute blocks or less. That means that we could probably squeeze in about 16 different topics for in the course of our 90 minutes and still attend to some housekeeping things. Of course, we could go a little bit longer if the discussion is very fruitful, that's okay. But we want to try and get in as many as we possibly can. So for panelists, as opportunity permits, we'll turn to you for some brief questions. I'd like to introduce them now. For those who have joined already, we have Walter Rauch, who's a contributing editor at StabilityHub and retired from AbbVie. He'll be handling a lot of the technical aspects of our meeting today, but he'll also be answering questions. Jeff Carr, also a contributor to StabilityHub, retired from Patheon, and he'll be tackling some analytical questions. Maria Krish, who's with us from Freethink Technologies, a whole lot about ISO conversion and handling stability in a newer way than we're used to about 30 years ago. And we may see representatives from Apogee Therapeutics. Nick Piscatelli might drop by. We have questions about that. Susan Cleary from Novatech and Pedro George from Vitatech. Health would be able to answer questions about stability systems. And we may see a chamber rep or two, and Mike Lally from Lighthouse Instruments, who's going to represent laboratory equipment that his company does and answer some questions about the latest in that field. So welcome to all the panelists and welcome to our participants. I kind of feel like I'm hosting a debate here, so having seen a lot on TV these days. So let's start off with a question that came through just yesterday. And it happened to line perfectly with some of our panelists and experiences that we've had. And their question was around a question they got from regulatory authorities who noticed that some of their products had repeated excursions in their chamber. And this company had done all kinds of studies. They did accelerated studies. They did let's see, cycling studies, they did some shipping studies, and none of that seemed to satisfy this regulatory question. They wanted to know, can you accurately represent what happens as a result of multiple excursions in your chamber? And this question's been around for many years. I remember it being asked about 2 decades ago, and there wasn't a lot of great answers. The technology was a little bit lower back in those days. But I recommended that they check out several things to be able to do. But rather than go into all that, I'm going to ask Maria Krish about how to take care of those kind of things. through ISO conversion would be one way to do that.
Yeah, thank you. So as John mentioned, I'm from Freethink Technologies, and what we're probably best known for is a software approach to accelerated predictive stability. So there's basically the core of this is short-term experimental models based on a moisture-modified Arrhenius equation that are used to predict shelf life in various types of packaging. So there's different approaches, but I think we're the only commercial software available to do that. So we both do those studies, but also sell the software, ASAP. And so the approach, if you're using one approach to this, is to use an accelerated predictive stability model to look at excursions. And that basically the model has two variables. It has temperature and relative humidity. And the idea is that you're predicting, you get a continuous model of stability over time. If you have long-term data, you can then compare the long-term predictions to the model predictions to the long-term to verify it. And some companies base their SOPs for excursions on basically on this model. So if after the excursion, you're still passing with a certain criteria, usually it's 95% probability of passing, then it would be considered acceptable. And because it's basically a functional dependence on temperature and humidity, you can add as many temperature and or humidity excursions as you want. And basically the basic model stays the same and it can accommodate that. If there is, I don't know if the person who asked that has those software capabilities, certainly feel free to reach out to me. I can kind of show you how to put in the excursion, but it's flexible. You can put as many as you can. So it's not necessarily making a statement that there's no impact of the excursion. It's basically evaluating how big the impact would be. And it's based on older testing, which basically has a very large temperature and humidity range. So usually the excursions are well within that sort of experimental range. So I don't know, that's other.
Yeah. Any follow-up questions to that, or anybody who's had experience with that particular challenge and found something that was successful with the regulatory folks? Okay, not seeing any of that for the moment. I'll ask a follow-up question for Maria, and then we see that there's one in the chat lined up as well. Follow-up question is, how successful is ISO conversion results going into regulatory authorities, aside from the method we just mentioned in a broader sense?
They're used in a whole bunch of different ways, depending on the kind of stage of the product. So I see there's a question in the chat. This particular approach, you do need software to do. I think that might be a sort of follow-up question for other folks. I will say that the accelerated predictive stability is not exclusively used for excursions. It's used to set clinical shelf life, sometimes early stage. It's actually ASAP. It's called, our program's called ASAP. ASAP only data is used, and often to support various sort of late stage changes in products. For NDA inclusion, that's usually what it's used for, to sort of establish things as a minor change, do comparative studies, also to support specification requests. So it's used in a lot of different ways depending on the product development cycle. So we definitely have seen cases where it's included in regulatory filings specifically for excursions and maybe only for excursions, but it's also used to kind of support packaging to get to the clinic faster and things like that.
You see, there's also a follow-up about what is applicable to mAbs or other biologics.
So not as a blanket thing, no, it is sometimes and not always. So I think once you get to larger systems, it's a little bit more case dependent. It depends on what is limiting stability and how precise the analytical method is. So typically you need an analytical method that's about 20%, no more than 20% error compared to the starting to the specification limit. If it's bigger than that, you can get a model, but it doesn't give you much. For larger systems, there's actually a fair bit of literature lately about people's experience using it. So I would say I'm as you mentioned, I'm at Freethink Technologies. We have been trying to collate some of the recent papers that both us, but more our users of the software have published. Some of it is in that area. So there definitely have been some cases of larger systems, but again, it depends on what's limiting stability. So there are cases where it's not appropriate, but there have been a lot of cases where it is. So.
Thank you. And then if it's not the case, then you're back to justification through excursion, accelerated studies, shipping studies, those kind of things where you make a case that you've gone way beyond. And that's had some limited success in explanations before. Let's go back before we lose it. Any updates on the Q1A R2 revisions as of today? Over the last couple of years, I've heard a lot of activity in this area that is being updated, but I have to confess I haven't seen anything recently. Do any of our panelists or participants have any updated information? I know surprisingly you'll find a lot of folks coming out of the woodwork that serve on these committees and panels and are well aware of exactly where they are these days. Looking for raised hands. Okay, so we'll have to leave it that it is very active. It was coming along very quickly. I presume it's moved much closer than the information I have, which was last June, that it had made amazing progress since the year before. So it is coming, and sorry to say that we don't have a lot of information right here, but that's a good question we could start tackling for StabilityHub. Go find an article that's given us an update to do that. So we'll get on that as a question. Let's see. This morning, I scanned speaking of StabilityHub, scanned all the incoming emails and saw an article that looked pretty fascinating about the impact of water in the atmosphere of your products and how it permeates our packages and the impact of that. This particular article had to do with microbial impact, but in general, moisture has a detrimental impact on stability all along the line. We have Michael Lally from Lighthouse Instruments with us today, and I asked if he might comment on that a little bit, give us a background on the technology, what's available, and why that's important for us.
Happy to help, John. So there's two USP chapters that are very important. USP 1112, which is the impact of water activity on the growth of microbial organisms in non-sterile pharmaceutical products. This chapter has been in place for about 15 years. It's been led by a team of microbiologists who really were kind of at the forefront of looking at water activity for oral solid dose. And the second chapter that's extremely important is USP 922. You can kind of view that as a compendium method. How do scientists in the pharmaceutical industry measure water activity? So there's two USP chapters. John's referring specifically to microbial growth. So if I have pills, tablets, capsules, you can also extend this to food. There's a certain amount of water activity, the amount of free water or available water that will support the growth of organisms. And what many pharmaceutical companies like about USP 1112 is it can be part of a risk assessment program where you can substitute water activity testing instead of microbial limit testing. That's pretty popular in pharmaceutical companies. The second big area is chemical and physical stability. Maria and the team at Freethink Technology has done just spectacular work looking at the equivalent relative humidity inside a pharmaceutical package and how that impacts the stability of a drug. So all of these different technologies just simply require an accurate and precise measurement of water activity. And if you have questions, Lighthouse can definitely help.
Okay, thank you, Mike. Any questions following that particular topic up? Okay, also something we'd like to kind of get into more in Stability Hub to cover that area. So we'll be talking with you about that. Be nice to have that as a permanent record in our archives, in addition to this video. Okay, let's continue on with some other topics. We had one submitted earlier. What tolerances are applied to pull dates for stability samples, and are timelines applied for start and completion of sample testing? And that's a popular question. People want to know a lot about timelines. I can kick off that discussion and see how many other people want to chime in. And that's that, yes, there are tolerances applied to those. They do vary, particularly depending on the day and the tides and the moon for the inspectors that are walking through your laboratory. Some are quite generous and apply wider tolerances. Others are very conservative. The most conservative is if you pull and send the sample on the day it's due, which of course it's a hardship on weekends, holidays, and busy times. Other people are mildly conservative and say within a couple of days they'll do the pull, and usually that flies totally well. Other people, and have done this successfully, they'll justify going out a couple of weeks, if not longer, depending on what phase of the study it is. The longer further out, the more tolerance they give on the pull dates. And some have gotten away with that just fine, and others have been called and have to move back to something more conservative. I'll stop there before I cover every bit of that. Anybody have other aspects of that they want to ask? Perhaps suggest a range as to what you might like to try, and we could comment on.
Yeah, John, can I jump in on that one a bit? It's a question that comes up pretty frequently in stability workshops that I've been involved with. And you're absolutely right. There's all sorts of different approaches that can be applied. Regulators have their own views, which may be variable, probably are very variable. I would say the most important thing here is that whatever it is that you decide to do within your company, you have to have an SOP that defines that this is what we do, whether you like it, well, whether you agree with it, whether you don't agree with it. This is the way that we've decided to do things, and you also need to make sure that you do have a lot of evidence available to show that that's what you have been doing. There may be occasions where you aren't able to achieve what your SOP says. And if you do that every time that it happens, you have to have a deviation record, which says what happened, why it happened, what you're doing about it to prevent it happening again. You also mentioned that you might vary the tolerance depending on the stage of the stability study, which does seem to make pretty good sense. At Thermo Fisher Patheon, this is certainly something that we did discuss. In that company, we were doing several hundred stability studies at any one time, and I would be careful of that approach. We decided not to apply it because you're going to make mistakes. As things go from one phase to the next phase, you're going to make mistakes, and then you're going to be into deviations as to why it happened. So we prefer to keep things simple. And just to put some numbers in there, the sort of thing we applied was that for pull tolerances, we allowed ourselves a 7-day tolerance on the pull date. This was to allow for things like weekends, national holidays, stuff like that, or other things that might happen. As far as testing is concerned, our requirement was that commencement of testing must happen within 2 weeks of the pull date, and completion of testing must happen within 1 month of the pull date. So for a ballpark, those are the numbers we used, and we applied the same figures regardless of what time point we were at to within the stability study.
Okay. Thank you for that input. Any others?
Yeah, John, I guess my experience has been that you can justify pulling early with your long-term condition, but not with stress conditions. At one point, we did a percentage, but then changed it just to a standard number of days, which was easier to manage at that point. But we were never challenged about pulling, say, a week early on a long-term condition. And again, I agree completely. You need an SOP and follow it.
Mhm. Thank you. I once worked for a company that was consistently late in getting their samples pulled and tested within 30 days, and they got a 483, and a year later they were asked the same question and hadn't developed fully into tackling that in its entirety. So very serious problem if it's a consistent kind of thing that you're chronically late and then haven't put the resources into fixing that. And you get to define what late is, as John just mentioned, as long as you have the SOPs and some justification for that, like a long-term study. Okay, I can bring up another one. Kind of related to testing, what are the requirements, or industry standards for a stability organization to review the raw data submitted to them from a contract lab? And Jeff, this might be up your line or some of the others that are listening. Requirements or industry standards. I think it's more industry standards if I'm not mistaken, but I'll let you comment on that, Jeff.
Yeah, okay. Yes, that's an interesting one. I guess it might also depend on what you actually mean by the raw data. If you mean raw data as I would expect it, it would be if for let's say HPLC methods, which is what most of the methods are. We were using Empower as our software system. The raw data would mean that you have to go into the Empower records, go through all the audit trails and all sorts of stuff like that to make sure that everything had been done correctly. And certainly clients were never invited to do that sort of thing. Regulatory agencies certainly did it, but clients would not be allowed to, on every delivery of stability data, we would not have clients going through our Empower records in order to verify everything was okay. They were invited to do audits, which they certainly did from time to time. And when they did audits, then they were allowed to, their auditors were allowed to go into Empower. We'd have a laptop set up where they could go into Empower and just see their own records when they did an audit visit, but they wouldn't go through every delivery of results to do that. So in my experience, it's not something that does regularly happen.
Okay, thank you for that. A bit of history there. I once worked for a company the one that was chronically late, was asked when they had use of a CDMO if they did 100% review of all the raw data as defined by the FDA at that point in time. And they said that was an expectation, and we fought back against that. We were not aware of industry doing 100% of raw data. They could do 100% during an audit for a segment. Other ways that you would do it, and you would set it up in your SOP exactly what would be your standard to do, and then follow through on that. So in this case, they gave that answer back to the FDA. I'm not sure whatever came of that. Wasn't with the company much longer afterwards. But there are some times where that gets challenged, and it's perfectly okay to push back if we have some justification as to why 100% is not necessary. In fact, it would take so much resource to do that may not be practical at all. Okay, welcoming other comments. If not, we can move on. There's one about stability software study management options that are suitable for small stability programs. Not necessarily a statistical package built in, but it's something to track the study samples and results. We're aware of some very large companies that do a fantastic job with that. There's also some ones that specialize in the smaller companies, as is mentioned here. And it's not our policy necessarily to endorse any one company, but we're just wondering if anybody had experience and can name a few.
And John, I've just gone through the exercise of implementing a stability software program, and having used one particular vendor for many years in the past, looked at them as well as anything else on the market because I'm with a much smaller organization now. Did not find anything that really is tailored to a small organization. Everything that you're looking at, it's probably, over 100,000 for year one to implement. And so I agree with the on the question, what else do you do? And what we've been doing for the last 4 years or so is just managing it through Excel, Excel and Word documents to generate summaries that way. That works for early phase development, but you're not in a validated environment. So that's the drawback. But I'm not aware of any other lower cost solutions.
Okay, thank you, John. Any other input? Okay, that continues to be a search then for vendor opportunities for somebody who can come up with something satisfactory and relatively inexpensive. But it does take a while to implement those kind of things.
I did have a follow-up on that. I was very interested to hear you were in Excel because I know that's a very easy and practical solution for small stability organizations. And in the past, we've adopted or kind of brought in some small programs into our larger program. Mike, I'm wondering if you saw or have experienced any pushback from auditors, at least, especially from agency, but anything in the industry that would say that is a risk. I mean, if you're handling all the risk and doing it right in Excel, you should be fine. But have you experienced an auditor question why you don't have a more robust or
Yeah, we have not yet. We're into a phase 2 clinical trial, so we're in the IND, IMPD state right now. So we're early on, and I think for the organization to invest in the software at this point makes sense because our current data is not so extensive. I think within about 2 or 3 weeks, we'll be able to bring it all into our software once we're validated. But it's the practical versus the economics. And if I had a solution that was 30,000, I probably would have taken that now, but I couldn't find anything.
Has anyone else experienced a question, some concern from an auditor, not necessarily an observation, but just concern there? Okay.
If we don't have an answer to that, there is a follow-up about, are you still using a separate inventory control system while you manage the studies in Excel?
And I can answer that for us because we're a virtual company. So our stability studies all are at contract organizations, and we let them manage the inventory quantities. I don't worry about that because it's specified in the protocol on what was set down on stability. And in a previous role in a much larger finished product solid oral dose organization where we had 500 stability studies ongoing at any given time, we used a paper system rather than electronic. So when people pulled samples from the chamber, they wrote it in a logbook and listed how many remained. And to me, that worked very well. And it's a case where low technology was much easier.
Mhm. Cost is a big factor when it comes to that. If you do that in a much larger company, then yeah, it's kind of high risk, and it's a little bit lower risk in a small company. But until you grow to those levels, it's the only practical answer, and it's a legal answer as well. There's no problem with doing that at all, just as long as you're diligent in keeping those records.
And one of the keys was we did an annual inventory check also to
Mhm.
verify. And just to comment other things that we did to manage the inventory, we had, when we put samples into the chamber, we put a color coded sticker that had a place to put the time point and the date pulled. And so that did a couple of things. And that this actually goes way back when to when ambient was used as a storage condition, as well as 2560 and some other perturbations in there, that to discern where the sample had been and how it was stored. And you could tell at a glance by a different color label what condition that represented. And then that bottle sitting in the lab now can be identified of when it was pulled, who pulled it, and what time point it represented.
Thank you. That's a good, thorough answer, and also a great answer to another question that was asked. I'll ask it kind of retroly because there was a follow-up to it. When samples are labeled with basic information such as product ID and batch ID prior to the initiation of a stability study, do they need to be individually labeled with stability labels as well? And I think you mentioned why you would do that to give it better identification as to where it's coming from, opportunities to record information on it. If so, do you pre-label them with the test intervals? And that's a great discussion that's been taking place for 40 or 50 years. I've seen it done both ways, seen people do it successfully both ways. To me, it was an invitation to, if you pull the wrong sample, then it's misidentified as to the interval that you're sending. So there's a risk there if somebody's not careful when they pull the sample out, as opposed to just pulling one randomly and then writing the information on it afterwards. So yes, it's been done both ways. Test intervals there's reasons to do it and reasons not to do it. So again, justification, SOPs, and watching what's happened to other people. If they ran into some catastrophic failures along the lines of quality that way, then don't do it. But if you control well, then you have no reason to change.
And just to add to that, John, something that came up, but we never saw an issue, was a concern of the adhesive impact permeating through the container into the product. And we never saw an issue. This was mainly solid oral dose with a few cough cold liquid products. But we never saw the permeation. And so we never worried about it. My preference was always to put like an Avery label on top of your existing label so that it would have to permeate through that label first. But we never saw an issue.
Yeah. No, I think that's a good point. Way early in my career, I saw a whole neosynephrine nasal spray study go down the tubes because the ink penetrated right through the softer polyethylene that we were using and created a cloudiness. So yeah, good to evaluate what's the impact of your label on your product. Are you using approved glues and anything along those lines? And especially even if you're going to write on a label with a marker, that marker material often permeates through as well. Thank you, John.
In a follow-up to that inventory question too, a real significant issue with inventory would be data integrity. You could do it in Excel, but you would need to somehow lock that Excel sheet down to track and have an audit trail and some sort of password, et cetera, protection for specific users. So it'd be a whole lot easier to have some software specifically for inventory tracking, in my opinion.
Mhm.
And that's why in my previous life, we went to paper or kept paper,
Mhm.
never advanced the technology. And the paper also is a good way for auditors to look and see are you pulling things on time or not. So
Yeah.
it's a double-edged sword, but we used it as an internal method to verify that everything was being pulled at the appropriate time point.
Yeah, this is a perfect opportunity to put a plug in for last month's topic, digitalization. If anyone has more questions on that, that was the topic that Novatech sponsored last month, and we had a similar webinar on that. Not disagreeing or agreeing, just pointing out the task of converting from paper to digital, and then to further digitalize and make it a lot more accessible and usable data digitally.
Mhm. And the recording for that is online in StabilityHub if anyone wants to go back and view that webinar. Let's see, I see a very long comment suddenly appearing in the chat. I got to scroll back up a little bit. How are those in the CGT industry conducting stability studies on the final drug product, particularly when the manufacturing process produces a limited number of final containers? For example, 1 to 2 maximum. Do you produce smaller side samples for testing? If so, have you received regulatory pushback for the side samples being in a different container configuration from the final product? That is an excellent one, and I don't think we have Nick Piscatelli on board with us today. He was invited specifically because he was a member of a therapeutics company. And these are the questions that are plaguing us, and they're relatively new, so I can understand exactly why they're coming up. So we can speak to that generically, unless somebody else is in that industry and wants to comment. They have the exact answers on these things. Because CGTs are very short-lived and usually made in very small batches, that poses a problem. And I would think that if you have SOPs and justifications on how the large package and small package produce similar results, you have a good leg to stand on. So I'm not aware of regulatory pushback yet on those kind of things. I think that I have seen some articles, though, that the FDA has been kind of understanding that this is a new area. The challenges are different. And I think I've seen people say that they've been lenient in those kind of things, whereas we couldn't get away with that in the other aspects of our industry. Anybody want to comment? One of our goals is to identify people such as Nick and now maybe our questioner as those who are pioneers on this aspect of stability, and we can continue to watch their experiences and what they've learned, what kind of feedback they've gotten from regulatory agencies would be very helpful to the rest of us. So my apologies that I can't go deeper into that. That's as far as I know. But justifications, as far as you can go, and procedures so that you're consistent and based on science. Okay, I'll jump over to follow up to the previous question. Has to do with sample control and movements. When our lab receives and stores room temperature stability samples pending testing, they are not held in a mapped or qualified environmentally controlled space, but simply in the regular lab area that is controlled by building maintenance. Is there any regulatory guideline dictating this sample storage situation? And I would expand that, just not room temperature, but if you pulled a sample, say at 40 degrees, you don't necessarily want to have that stay at 40 degrees too long, because it's no longer 40 degrees at that time that you specified. So it needs to come out and do something rather innocuous. My experience is that I've not seen anybody get a 483 based on that storage unless it was extreme. it'd be a great idea to monitor that area, even though it's ambient, just to show what the range of the temperatures are in. It's not really an excursion from a laboratory condition. You need to keep a log of what's there. And we actually moved samples from all kinds of conditions into 25-degree holding chambers, so we knew that we had it there rather than in the laboratory where it would vary. But I don't see any regulatory kickback on that kind of thing, and it takes up lab space when you've got samples in there, and then the FDA starts asking, why is a 40-degree sample sitting in your 25-degree chamber? So that can raise its own issues. But I would say got an SOP justification that it's not large deviation from your laboratory conditions, or 25 degrees if that's where you're trying to store it at, and that it's controlled well. Anybody else want to comment on that?
Yeah, John, if I could. Yes, it's right. Your laboratory area is not going to be controlled the same as your stability chambers. However, you are supposed to monitor the temperature and relative humidity in the lab areas. And when you have regulatory audits, it's quite likely that they might want to check the record for the what the temperature condition was at the time a particular stability time point was being held in the lab. And you should have that information readily available. You mentioned accelerated samples, but we would also keep those at lab ambient temperatures. But the other thing to look at is subambient storage conditions. For samples that have been refrigerated or held in a freezer, we would keep them in a fridge or a freezer up to the point that they're being tested. They would have to be kept under those sort of controlled conditions.
Mm-hmm. So ambient for that would be accelerated in actuality.
Yeah.
It just occurred to me that that also might be a reason that you'd want to have that monitored. If you had a failure in your laboratory investigation, would want to rule that out as a factor for the deviation you saw. I see Maria and John both look like they're, no, now it's only John looks like he wants to speak.
Yeah, I'm now over in the biologics world where we do store a lot of materials subambient, and you need to, so when those stability samples get pulled, they, depending on which lab is doing the testing, there are some labs that will thaw it and then aliquot it out for each test, and maybe store at still a below ambient condition. A lot of that depends on the material and how the stability of it. And you need to have freeze thaw data to also support if you're going back and forth, freezing and thawing. And that's where I use the glass transition temperature, which is much higher than the storage condition, but use that as a point to justify that anything below that temperature, you're going to have very little movement and subsequent degradation. But again, you need to specify in your documents how you're sampling, if you are aliquoting, and how you store samples prior to testing.
Thank you for that input. Just had a flashback as well to a couple decades ago when there were no biologics and people were pulling things out of their stability chambers at say 4075 and 2560. And it made perfect sense to them that they didn't want anything to change. And so they would take their samples down and stick them either at 5 degrees or the freezer. And that quickly got them in trouble with FDA inspectors because they needed to know that the refrigeration or freezing didn't induce other problems that the product might be susceptible to. So while perfectly reasonable for biologics and those kind of things, we discovered early on, and I think my 25-year-old self was one of those who used to stick things in the refrigerator before they went to the lab. And one day I walked down because people were complaining about samples were lost or never delivered to them in the analytical lab. And I said, well, where do they land when you bring them back to your place? And they walked me into a large refrigerated room that was used for storing reagents, and I could smell bromine particularly in that room. And there was a stack of samples, literally a pile of all stability samples, so just put on a circular table. And the analysts would have to go in and try to sort through that pile and find the samples that they needed. So we quickly revised that we held the samples where we were until they called for them. And that was how we got around a large sticky problem there, particularly because you could smell the chemicals in the samples once you opened them. So always be willing to walk down the hallway and see what's going on the other end when you deliver a sample. Where is it going? Who's handling it? What are they doing? Is it going to be embarrassing for an inspector to walk in and see what you just saw?
John, my recollection is, and this may have been the original stability guidance from the FDA that expressly stated that you were not to store stability samples in a freezer until they were tested.
Mhm. Wouldn't be surprised. I've not read that one lately, but I'm sure there's something in there. Speaking of chambers, does anyone have any experience with an investigator having a concern about chamber cleanliness? If so, can they share their insights? And again, we don't want to do all the talking, so I ask other people if they've got some experience about chamber cleanliness.
One of the issues, particularly for higher humidity conditions, is you inevitably get a little bit of salt if you're using softened water. And so we used to wipe the chamber down periodically, and I don't remember if it was quarterly or every 6 months, for just to make sure that there was no accumulation. But definitely you need to make sure that the chambers are clean.
Yep, and that yells SOP to me that you can specify how you do that and how often. An experience I had was that we said we wanted our chambers clean, so we told housekeeping that we want those chambers cleaned every couple of weeks. But being young and stupid back then, we didn't specify anything else. So what we discovered was that the house cleaning committee would go in, they'd throw back open the door, pin it back with a bucket. They would take a mop and swab down the floors and the walls, and the humidity rose to 1,000%. And the place smelled for a while with the cleansing agent. So we had to put a stop to all that and then go back to, okay, what are the industry standards for that kind of cleanliness? What microbes are you trying to kill? What levels of dryness do you need to prevent moisture from going into promoting mold growth, and anything along those lines? So yeah, definitely don't turn anybody loose to clean your chambers without an SOP, and maybe you watching them for a while. And what I discovered was if you're in an organization where the night crew never sees the day people and the people that they serve, it's a good idea for a stability manager to hang around some evening and do a little bit of an appreciation session with them, maybe bring in some sandwiches or some desserts or something, and give them a little presentation about what's in those chambers and the importance of their role in preserving those. And they take a great deal of pride in knowing that when they see the TV commercial come on for a drug that's been in their stability chamber that they cleaned, that's just as important as the person who discovered the product, because it never would get to market without those chambers. So that goes, a little bit of praise and instruction goes a long way in prevention along
Yeah.
those lines.
That's a
And
great point. Sorry, I had a situation where we were competing essentially with the maintenance people for the R&D side, and the R&D people told them our studies are most important, and we had to took them aside and said, look, this room has $5 million worth of product. We have the FDA on our backs. We are actually more important, but it was just like you said, having a good conversation with them, a good rapport, help them set their priorities, not to really cause a conflict, but to really understand that just because one group insists they're most important does not, they need to follow up with that and make sure their priorities are straight. That's good.
John, as you were mentioning things like mold, reminded me of just being cautious with cardboard in a chamber that to make sure you don't end up having a lot of microbial growth. We had it was a product in a metal canister that practically to store them, the easiest thing to do was keep them in the cardboard that they were shipped to us in. But at any accelerated condition, we actually took it out of the cardboard and put it into a plastic tote to avoid any microbial growth.
Okay, and we have some chat comments and questions. Someone once saw slime build up that blocked the steam outlet and caused water pour back into the chamber, opening the top of the chamber to check the steam outlet wasn't originally part of the maintenance SOP. And that sounds very familiar. I think Jeff is working on an article and cited something that happened along those lines. And you'll see that article in the November edition of Stability Hub newsletter. But yeah, that's a very valid observation. The slime can grow in your pots that are holding the water. You could also get moisture building up on the outside of your window when there's a high difference in temperature from the inside to the outside and the window is not heated. So if you walk by a stability chamber and see a little bit of rust around the side of the window, that's what's happening there. The other one was more of a question than an observation. It's thanks so much for setting this up. Wondering for years about refrigerators and freezers used for stability. In general, high performance or medical grade units meet the temperature control requirements well without causing my heartburn. Is there any industry preference for the units designated? Let's see, scroll this down a little bit. Designated as stability refrigerators or stability freezers? How are defrost viewed? And also, what do people do when confronted with storing flammable materials and stability? Are there good options before constructing a custom room? So 3 parts in there. Let's start off with people having any preferences. It's nice, I think, to hear from people that have experience with chambers that they purchased that were done by companies that are experienced with the needs of the stability world. So I think there may not be so much a difference as quality as there are in setting up exactly according to the needs and parameters that is required by stability might be a little bit different than just straight medical refrigerators. One of our participants from years gone by described he was an expert in chambers, but not necessarily stability chambers, and he received a request from the stability group to get a refrigerated unit. And he did, and had it installed, and within a week, it was the refrigerator that was serving people lunches in the cafeteria because he had completely missed all the requirements that were needed. So he's become an expert in that now. So we really appreciate his growth in that area. So that's my observation, is that if you got something that's installed by a company that's experienced in the needs of stability, that's an advantage over just plain refrigerators, even though they're technically good. How are defrosts viewed? This is something that I think they do less of, at least on an automatic basis. We used to have a refrigerator, frozen chamber, and it would defrost every so many minutes, and it gave you a horrendous output on your monitoring that the temperature was always changing. It was a quick change, but we often asked, well, why do you do that? And they said, well, you have to do that in order to hold down the defrost cycles, so you wouldn't get frost formation in there and eventually a lot of ice. The more modern chambers, I think, remove humidity, if I'm not mistaken. But anybody want to comment on that for defrost?
Definitely needs to be addressed in the qualification and mapping package. What the frequency is, and ideally in the midst of the qualification, at least one defrost cycle occurs. You need to be able to show it's not causing a spike in a particular portion of the room or something, or the chamber. Now there is the option, sorry, of just shutting off the defrost and having a process or procedure to essentially remove material from the chamber every 6 months and let it defrost itself that way. But that's, I don't know, that can be done, especially for lower temperatures.
Yep, that was my comment was sometimes it's induced defrost that you set it up, maybe you move your samples to some other place so that they don't defrost themselves or thaw themselves and then go back in and set it all up again. It's almost like a requalification kind of thing for the changes that you're going to make in order to defrost it. So yeah, I agree. That's definitely a way you can go. It's kind of elaborate in doing that. So maybe finding chambers that don't require defrosting or they're considered frost-free in some respects, realizing that if you artificially change the humidity in there, you don't want to do extreme dryness that will impact your product or your packaging. The other part of this has to do with flammable materials. I know sometimes chambers are required in certain states to have sprinkler systems in there. And while we talked about, John mentioned the mold thing, and also with cardboard, you could get paper lice, and that's a terrible thing to have as well. But inside the chamber, you can use plastic to get away from the cardboard. When I was working as a consultant once, and another company had just set up brand new stability chambers, they took our recommendation to use the plastic containers. And then they had an inspection by not regulatory authorities, but by the county fire inspector. And he cited them for having flammable materials, i.e. the plastic containers. I don't think they were capable of bursting into flame, but if you held the flame up to them, you probably could melt them. And they had to get rid of all their plastic and put in metal containers, much like you see in the micro lab with samples going through. And that was a real hassle trying to get your samples handled as easily with those kind of containers as you were the plastic. So just a word to the wise, if you're about ready to switch over from one system to the other, check all the inputs you possibly can, including the fire regulations, whether or not you need sprinklers, whether or not you can use plastic, those kind of things. I would imagine that there are standards for flammable materials, maybe extra ventilation to take off any vapors, that kind of thing. But to me, that means you're also going to impact the function of the chamber. Could potentially be cooler than what you want it. So why I don't have a good answer for the flammables. Anybody else have experience for that?
I worked extensively with flammable organic solvents, but they weren't part of a stability study. So I think the one thing, if you're truly dealing with flammable materials, you need your health, safety, and engineering people involved. Then they have to look at flash points and concentrations. Again, the question doesn't have the details, but if you're storing truly flammable organic solvents, there are some very specific requirements for that. It's very different. John's story about either cardboard or plastic is maybe a local fire department, but organic solvents and organic gases that are flammable, like if you're dealing with hydrogen and hexane, it's a different world.
Yeah, definitely for a problem to try and solve and still maintain your science of stability in that. More questions rolling in here as well, kind of related to all this. Is it necessary to clean evaporator units in 5°C chambers to resolve mold issues? I would think so, but the person to ask are your maintenance people. Good idea to have SOPs that involve that, and particularly with regard to preventive maintenance. You definitely know how often you're going to need to clean an evaporator unit if that's the case. And then a record of how often you found mold. If you've not found it at all, maybe you don't have a problem, or maybe you were just lucky, but you want to clean your evaporators on a periodic basis. So we're open for comments on that. How do we effectively clean out the chamber parts that are contaminated with slime growth? There are agents that are approved to do that. And whether or not you want those in your stability chamber in close proximity to your samples is a matter for some internal examination to see what's going on there, what's compatible, what might pass through your materials. If you have an extractables and leachables study going on, maybe you could throw that into the study and see if those kind of things are permeating your your packaging or your product or anything else along those lines. Other comments on slimes and molds and yeast and creepiness? If not, I'll take another one that's posted here. A question about drug substance stability. Is it always stored in the original containers of the drug substance? Is there any case that the drug substance are stored in uniformed containers to help with the storage space? I have some minerals that they consider as drug substance, and they come in big drums and we can't fit in the humidity chambers. This humidity chamber required for drug substance storage, as long as we have room that has the temperature and humidity monitoring daily. Good enough. Okay, a few questions in there. The answer is yes, you can use other containers, and we've done that quite successfully. It's supposed to be justifiable to use a bigger container of the same kind of materials and maybe the same proportions of headspace, same kind of plastic that you would use inside. It's been done, and people have stretched that one way or another from time to time. And I'm not sure that there's been any 483s that I've seen that somebody went too far abroad in those containers. We used to use mailing shipping containers to mimic the great big drums at one point in time. So I would say, yes, you can do that. And you want to have an SOP, you want to have justification. Humidity, I would say, probably is considered necessary for drug substance if you're doing an API study, particularly at accelerated conditions. You want to do that. But once you're commercial, I think that you could probably store it in the warehouse with monitoring, but I don't want to speak too far before we get some other input. So how about some from our panel on that question?
I can
John, we had
comment.
some. Oh, sorry.
Go ahead, Jeff.
Oh, okay, John, thank you. Yeah, I had some experiences of this sort many years ago. We were doing API, we were developing APIs, and we were putting them into stability studies. Typically, the APIs are stored in polyethylene bags, possibly double bags, which are then put in fiberboard containers, which I think is the sort of thing which is being described in the question here. The important thing for stability is to be using the primary container, and so the primary container is the polyethylene bag. And so what we tried to do was to get the supply of our commercial polyethylene bags, our large bags, to provide us with small bags which were the same composition. This didn't work all that great, and we did have some bad experiences when we were seeing apparent impurities turning up in the stability samples, which were never seen in the main containers. It turned out it was tert-butyl phthalate, which is constantly used in plastics, and these were in our small bags, but were not in the larger bags we were using. So this wasn't working. Although we asked for the same composition bags, we weren't getting them. So what we ended up doing was to take the large container bags, which were used for batch materials, which could be tens of kilos, and we cut the corners off. So you end up with two cone-shaped bags from the corners of those bags, and we took a whole set of those, and we used those for our stability samples, and then that way we were getting a good representation of stability in exactly the same containers as the full batch was going to be stored in.
Okay, thank you, Jeff. John, your comment?
Yeah, and just to add to that, when I was in solid oral dose, we actually had the fiber drum manufacturer make what we called a hat box, just a small, about 6 inches high, maybe 8-10 inches in diameter for us, that we used to mimic, and then we were able to get the same bags, same thickness, just in a smaller size, same thickness and material, smaller size that we put in. The biggest variable from between stability and the production environment was how well the bags were sealed and how that was done. They essentially went to a zip tie, and sometimes people would fold them back over, sometimes they wouldn't, but that was always a variable. And we we also used, if there was desiccant, we would put the desiccant inside that small drum in a proportional amount. There was always the question about the surface to volume ratio, and you can make arguments one way or another. It's going to be about the same, any way you look at it.
Okay. Maria.
Yeah. So as I mentioned before, for people, I'm not coming from a regulatory point of view. My company doesn't have much ongoing stability, but we do a lot of modeling of kind of factors that affect growth of degradants. And I think that last point that John Stewart made about the sort of scaling of the surface to volume and the fill is really important. So you will see for things that have significant degradation, in a lot of the bulk storage, most of the water uptake is going to be by the material itself, the drug substance or the product. And so if you really significantly change the ratio of amount of stuff to the surface area of the package, you may get results that aren't representative. So it's something just to be kind of careful about all of the sizing and the scaling, in addition to being careful about the materials.
Okay. Let me ask Michael, from the technical side of things. I know you measure a lot of things about the water and the headspace and the gases, and that's usually on a much smaller scale. Do you ever get any questions about what's going inside a great big commercial sized drum? And can you measure that?
Yes, we do. I really appreciated particularly Maria's comment. So one of the things that we've done to help people is a little bit different than your normal stability study. We're talking about the very large drums and the very large containers that are stored in environments where we monitor the temperature and the humidity in that environment, meaning a warehouse, a quality warehouse in a pharmaceutical firm, where we have time, temperature, and humidity. And these containers are going to be opened at different periods of time in order to feed the manufacturing process. Firms take a small sample at the time that those materials are drawn and measure the water activity and other properties, meaning you're going to be doing your normal QC checks for the purity microbial growth. We're going to gather water activity at the exact same time to know how much has humidity changed over time. Then some of our clients are using the FreeThink technology software, and they're able to say, if this product was stored at this elevated temperature and elevated humidity for this period of time, what impact did it have on quality? The water activity test itself is so easy compared to everything else that you're doing. It just gives you information that you wouldn't otherwise have to both predict chemical and microbial stability.
And to just comment on the water activity, this probably 10 or 12 years ago, we went and did a survey of all of our products for the water activity, looking for anything above that magical amount, and never found anything above that amount. And so we just kept the products on a monitoring program, just to continually demonstrate that we weren't going to have any microbial issues. Again, solid oral dose where sterility or microbial growth was not a routine stability test.
Any others? Okay, I'll introduce another one. Going back to some of the windows that we experience in stability for time. We have a test initiation requirement of 2 weeks from the pull date of a stability sample, but there is some debate on the definition of test initiation. Can you give me your opinion of what that would be? And that's a classic one that FDA has looked a lot at what the definition of this is, because if a company says that they're going to start testing within 2 weeks, and they simply pull out the sample and do a physical test, oh yeah, the color of this is good. And then they wait another 6 months before they finish anything else. That's kind of a violation of your SOP. So it should be a start and a completion in as well as to what it does. So it could vary. Some companies, if depending on the nature of their product, if they have something that starts to change right after they pull, say from a 4075. Now let's go the other way. Let's say it was cold and you warm it up in order to get it to the lab and need to thaw it. Some of their tests and parameters of their stability indicating tests will start to go downhill much faster than some of the other products. So you'll see some people say you got to start your test for X, Y, and Z in 2 days rather than 30 days, and then allow for other tests to be completed later on. But the idea again is nice SOP describing the process and why you do that. What's your justification? Why you let something go for a certain number of days and other ones that you require to be tested in order not to see any degradation that would happen while it's sitting around in the laboratory or waiting to get tested. Let's see, did I answer the question? I think I did. Walter, you got anything from your experience on that?
I do. Mainly from the definition of initiation, just like you said. I mean, I unfortunately had the experience of quite a bit of debate within between the lab management and us and the stability side of, wait, you started an investigation, and suddenly they would initiate, because of an investigation, would use that as an excuse for, we have this ongoing, so we can finish our investigation and test things a month later. And I don't know, it just seemed like, well, at the very least in that investigation, some mention needed to be included of the impact of that delay on the samples, at least somewhere. But it was still it was, unfortunately, the company I was in purposefully left it vague because they didn't want too much definition. So that, simply weighing the sample on the 30th, in this company, they had to have it initiated within 30 days. They could weigh the sample in 30 days, and that was good enough. Maybe, and maybe dissolve it in the solution the following day or something. It was just a very nebulous. Ultimately, what it came down to is whatever the analyst entered in the system as their test initiation date, which was a key part. At least there was that document, and that entered date was verified by someone. So someone did agree that it met the basic requirements of the procedure. But I have to admit, we frequently had concerns as to whether or not, concerns that it did not meet, in our opinion, the letter of the law, even if it was left intentionally vague.
Mm-hmm.
But I can't say, though, that I don't believe we ever got a observation, probably because an investigator never looked that deeply into exactly how that date was chosen.
Yeah, that's a good point. I always recommend that people include as many justifications for any kind of activity that they have. You could probably spend your whole career justifying every sentence. The other observation would be that the flip side of keeping things tighter so that you can impress the regulators that you got everything accomplished within 10 days instead of 30, or you pulled all your samples on time, is that you can tie yourself up in knots with the restrictions that you're not able to actually meet, even as admirable as those restrictions would be. If you don't have the resources to do it, or you have enough instances where you can't meet that, you're forever writing exceptions and deviations, that kind of thing. So it needs to be a happy medium someplace in there. Deeper dive into the laboratory side of things. How many significant figures are used for reporting of results for various test methods for samples, stability samples? Is it usually recommended to report results to the same number of significant figures that are used in the product specification for each test method? But sometimes a different approach could be preferred. How would we narrow that down? What's the thinking about the use of a certain number of significant figures? Jeff, do you have a comment on that particular one from your laboratory perspective?
Okay, yeah. Yes, that's it. It says there. The usual recommendation would be that in your specification for each of the individual tests, you have a requirement which is defined to a certain number of significant figures. So when you report the test for that for that method, you should report the same number of significant figures that the specification says. And that's usually a fairly good thing to work with, but in some cases, it's not great for stability. So for example, if we're dealing with related substances, and you have using an ICH limit for a new unknown impurity, which typically is going to be 0.2% to one significant figure, would you report to one significant figure there? And we would report to 2, because very often when you start the stability test, there will be something reportable or visible in the initial sample. Let's say it's 0.1%. And now you're going to record it. And if you only report to one significant figure, you're just going to be reporting 0.1 or 0.2, and then it fails. If you report 2 significant figures, then you can see a trend. You might be able to see a trend developing, which you can't see with just one significant figure. So in those cases, we would incline to report to 2 significant figures, even though specification was requiring 1 significant figure. So I was just wondering, how do other people deal with the same sort of thing, which is probably a fairly common type of situation.
It sounded like you were quoting from a guidance there at the beginning. Is that the ICH? Regarding
it's in the it's the Q3 guidelines, Q3A for drug substances, Q3B for drug products. So typically it's Q3B. Q3B for drug products, most of the recommendations are telling you to just report those for 1 significant figure.
Okay, and then with some justification, as you mentioned, it could serve you well to go to 2. At what point do you run into any problems with going too far? Is that described within the methods that are outlined? What's your limit? In other words, if you want to go to 3 significant figures, or your sensitivity on your tests allow that in all cases?
Probably not. Probably 3 significant figures is not a great idea.
Okay. Thanks. I just didn't want us to get carried away.
Yeah. But it also could apply to dissolution testing, where usual specification requirements are just whole numbers. So you don't report any decimal at all. It's just whole numbers. And for stability, we would report those to 1 decimal place. So that would be 2 significant figures.
Okay, thank you.
Yeah. Actually, it's 3 significant figures.
Any other comments on that before we move to the next question? Okay, Here's one that I would have thought was a crazy question about 15 years ago, but with artificial intelligence, it's quite possible today. So is there any software out there that will convert a scanned document of tabular data into something that can be put into Excel where that statistical software could use? Sounds like we're doing an automated digitalization kind of process. Has anybody actually seen that in use or read about that kind of equipment? Takes a picture, turns it into data, you can start processing. Okay, still kind of advanced for us. Probably pretty expensive to get to. But keep your eyes peeled for that kind of stuff. Then I'd be interested if it's data that's handwritten, how good is the handwriting that it's interpreting? Well, how do you validate that kind of interpretation?
Yeah, exactly. It seems like AI is getting to the point where it could probably do that pretty effectively, but to validate that would be a nightmare. Yeah.
That's the case. Yeah, handwritten stuff is quite interesting in the pharmaceutical industry. For a while, I was sidetracked from the stability area over into QA and was reviewing batch records. And if I looked down and saw some stuff that I found hard to read, I would take it down the hallway to the people who wrote it and said, I can't read that. Can you identify it? And they got quite ticked off with me doing that too many times. They couldn't see the point because everything turned out okay in the end. So I had to take a different tact. I took the character that I couldn't read, say it was a Q or something, and I would take it down to the copy machine and I would blow it up about 1,000 times till it looked like a piece of artwork. And then I would take it down to the person who wrote it and say, hey, can you identify what this is for me? And they'd say, I have no idea. And I said, well, I don't either. It's yours. They laughed and they started being real careful with their handwriting to do that. So yeah, human writing and recording is very difficult to validate and often can lead to some mistakes if people aren't really careful. Okay, beat that one to death.
All right, it looks like I don't know if
We
someone
got another one.
mentioned this already. Anita included a link there
She's
to
seen
an
it and she's
article.
given us a reference.
Yeah.
Thank you, Anita.
Very nice.
Cool. We will keep that as a permanent record here and make it available to all of our readers and viewers. Much appreciated. Another one. If a significant change is observed in an accelerated condition sample in the stability sample for a zone 4A territory, for example, a degradation product that exceeds the specs, does that mean that no shelf life extrapolation will be accepted as the zone 4A real-time storage condition? It's the same one generally used as applied for the intermediate, as per extrapolations noted in ICH Q1E. So a couple of parts to that, but the idea is if you're dealing with something that's plenty high to begin with, you're actually doing a zone 4A condition that's more of a real time than accelerated. Anybody have any experience with that or tried it and had a problem with the authorities?
In a past life with a solid oral dose product, which was stored at room temperature, we did not have good stability, either 4075 or 3065, and we were limited to using our real-time data from 2560, and it really hamstrung us. And we many times tried to submit something saying, oh, it's not predictive. And they'd say, well, that's fine. You need real-time data.
Mm-hmm. So good idea to be in contact with the authorities so you know that before you end up quote right up against the registration period.
Yep. But it made doing anything with that product extraordinarily long because you needed to wait 18 months to have any useful data.
Good observation. Any other input on that? Okay,
Yeah, John, when, sorry, when this thing first came out, was some years ago now, I think its guideline was Q1F, wasn't it? Which was then eventually withdrawn. But ICH were offering us the possibility to do our stability studies at 3065 instead of 2560. And that way we would have data which would be usable in possibly third world countries without having to do any additional work for it. But I think a lot of pharmaceutical companies, certainly a company I was working for, we rejected the whole concept of it, because if you did have a failure, then at your accelerated sample, you have nowhere to go because you don't have an you don't have an intermediate condition anymore. And so everything would have to be based on just real-time data. And so we didn't do it. And I was just wondering whether other companies still take that same sort of view or not.
looking for raised hands. Okay, doesn't sound like we have anyone that's knowledgeable in that particular situation to give us a response. So good question, Jeff. Hold on to that one. We might be able to put it up in the discussion area of Stability Hub and see if we get a response from a greater audience. Here's a quick one. Any recommendations on having a second person verify a sample pull from a chamber? I think it's a great idea when you're talking about sample control and verifications. Helps prevent errors as long as somebody's actually reading what they're signing off on to do that. And I think that we're starting to get to an area where technology can do that as well. It can take, for instance, electronically what's due, and then compare it with a scan of a label that's there, and verify that what's due is the label that you just picked up and scanned. But barring that, back in the subject of reality and smaller budgets, second person probably serves that area well. Second person when pulling the sample, and I would say a second person receiving the sample would be a great practice. Anybody want to discuss whether that's a great idea or over the top? But if you don't have two people in the stability room.
We had a forced case of doing that because of storage of controlled substance samples. So just to get into the area required two people. And so there were actually always two people there.
Very good. I see in the chat that somebody does secondary verification for all polls. So if you can, it's a great idea. So that's the answer to that one. One more here, and we're reaching our outer limits on our time period. Self-imposed, so people can get to their 12 o'clock meetings. Some testing is predetermined to fail at 40 degrees C, but has good outcomes at 30. Understanding the compound chemicals used in the product can give some leverages with the FDA, such as nifazoline impurity. So that kind of addresses the question we just had at the higher station. And also brings up the idea if you're scheduled to test at 40 and you already know it's going to fail. I think after you've had 1 to 2 failures, depending on your justification, you are justified in taking the rest of the testing at that higher station out, buying into the fact that you're not going to store at that temperature based on the data that you have. So with that, I think we need to do a little closing out housekeeping, and thank everybody that came and joined us and participated through listening or discussing, and especially to our panel members who shared their expertise. We do more of these in the future. Some will be generic like this to take any questions, and some are set up on specific topics. So as Walter mentioned earlier, we did digitalization. That was the first in a series of 4. And in November, we have one on stability chambers. That's also a series of 4 coming up. And we'll start out with not just chambers in general, but the kind of problems that occur in chambers with our samples and what we do with those in response to chamber failures, such as leaks and those kind of things that we actually had a mention of here in our agenda. As we go into the winter, we'll be approaching Stability Month and Stability Day in February. We'll have a special program recognizing stabilitarians. We'll also entertain questions there and have a nice program, especially if you want to share it with your constituents at work. And then we'll be back into another general session like this, Stability Hub office hours next May. And we welcome you to attend any and all of those sessions. We're really glad that you participated in this one. So with that, I'll say thanks and start to close out the meeting, and we'll stop our recording.
Thanks, John.